So again, the stimulus for the negative feedback loop would be a drop in blood oxygen or a drop in red blood cell number. The kidneys are the sensors. They're also the control center because they're releasing erythropoietin. Erythropoietin targets the red bone marrow and stimulates red blood cell production. Some athletes do this thing called blood doping, where they take artificial erythropoietin or they just get more red blood cells, and they do this so that they have higher oxygen carrying capacity, but that's dangerous because then your blood becomes too viscous. So that's just something to kind of put in your back pocket and be aware of. Erythropoiesis takes about 2 days to form your red blood cells, and then they're going to eject their nuclei, kill off most of their organelles, and be released to the blood. So as far as their life cycle goes, they circulate for about 120 days. Doing their job of carrying oxygen around. That's their main function. They can carry some carbon dioxide, but that's kind of like a secondary function. Their main job is to transport oxygen. So they circulate for 120 days, transporting O2 to our tissues. And when we look at capillaries, or the blood vessels, when we continue in the cardiovascular system, we'll see that capillaries are very, very small. They're the smallest of our blood vessels, and our red blood cells have to be able to squeeze through them. So they will kind of like fold up when going through a capillary. And when they lose their ability to do this, they're old and they're not really going to be able to function much anymore. So the spleen, and to a lesser degree the liver, but the spleen has test capillaries. So after 120 days, the spleen is going to kind of test them. So we'll say the spleen has test capillaries. And if they become rigid and can't bend and get through those capillaries, then they're going to be eaten up by macrophages. So we'll say if the red blood cell cannot squeeze through, then they're gobbled up by macrophages. Or we'll say then macrophages are going to phagocytize them. So this happens in the spleen, it also happens in the liver, primarily the spleen. The macrophages will gobble them up, so it would look like you get this blood cell that can't fold and bend through that capillary, so then a macrophage will come and gobble it up. There are some valuable things, though, in our red blood cell, so what do we do with it? We've got 3 different regions. We've got the heme portion that has the iron. So from the heme, we'll take the iron and we'll recycle it if we need it. So we'll bring it back into the system to make more, we'll take the iron out of that to make more red blood cells. The rest of the heme portion is going to become this stuff called bilirubin. So if you remember, the heme group was surrounded by that globulin protein, so our iron will be taken out of that and recycled. The rest of the heme will be converted to bilirubin. Bilirubin goes to the liver and gets concentrated in bile. So the liver will take it and concentrate it in bile. And then the bile gets added to the small intestine and ultimately removed from the system through the feces. So this is actually what gives feces its color. So concentrated in bile and removed. If we have excess iron and we don't need to reuse it, then we can also sweat this out, or it can be excreted in urine, or it can be excreted in feces. And then as far as the rest of that globin portion, so the globin portion was all that protein, so the globin will break down and recycle the amino acids. That's life cycle of a red blood cell. It begins with erythropoiesis in the red bone marrow. Red blood cells are mature once they've killed off most of their organelles and ejected their nuclei. They circulate in the blood for about 120 days, doing their job of transporting oxygen. And then when they no longer fit through those spleen capillaries, test capillaries, they get removed. Macrophages eat them up. So we take the iron part and we recycle it if we need it, or if there's excess iron, we can sweat it out or excrete it in the urine or the feces. Heme gets converted to bilirubin, which goes to the liver and then is concentrated in the bile and removed through the feces. So this is why those little babies whose moms are Rh negative, and you have an Rh positive baby and they're born with jaundice, they're really yellow, it's because they have a whole lot of bilirubin in the system. And the reason they have a whole lot of bilirubin is because their mom's antibodies are attacking their red blood cells, killing them off. So we get a whole bunch of this bilirubin left over, and that shows up in their skin and they look kind of orange. They get that jaundice. So again, the erythrocyte life cycle begins with formation in the red bone marrow. That's due to erythropoietin. These cells circulate for 120 days, and then they're degraded or gobbled up by our macrophages, and we recycle the parts we need, and we excrete the parts that we don't in waste. So any questions? That's kind of the just short version of everything that we just went over. Make sure that you can describe each one of these portions of the red blood cell life cycle. All right, leukopoiesis is the formation of white blood cells. And you always have low levels of white blood cells circulating around. The white blood cell's job is in the tissues, so most of their life is spent in the tissues fighting pathogens. Leukopoiesis is going to be their formation, which again happens in the red bone marrow. So this occurs in red bone marrow. And leukopoiesis is more complex. We've got different types of white blood cells that they're all going to start with that same hematopoietic stem cell, and they will follow one of two differentiating lines. A myeloid line or the lymphoid line. I'm not going to make you worry about the details of the intermediates. Ultimately, they'll become our neutrophils, our basophils, our eosinophils, our lymphocytes, or our monocytes. And what's going to determine that is what's going on in the body. So if you're sick, if you've got a bacteria, then there will be these chemicals called colony-stimulating factors. So we'll say colony-stimulating factors will guide cells along their differentiating pathway. So we'll say different pathways, different developmental pathways. And the colony-stimulating factors that are around are dependent on what pathogen you may be fighting. So white blood cells fight pathogens. So if you've got a bacterial infection, you'll have colony-stimulating factors that take that hematopoietic stem cell and guide it down the neutrophil line, and also down the B cell line, because our B cells are going to be able to make specific antibodies that fight that extracellular pathogen. Again, we'll go over all of this when we get to our immune system, but I don't think we're going to go over the details of leukopoiesis in great detail, because it's just, you have enough to worry about. And just being aware of what it does in general is what's important. As far as the life cycles, again, they are formed in the bone marrow. Then we store some in the spleen. So you store some in the spleen. And our secondary lymphatic organs, which we'll talk about when we get to the immune system. But their job is to live in the tissues and fight pathogens. So they live in mature cells, live in tissues, and fight pathogens. So you shouldn't have high white blood cell count in your blood really ever. If you're having an immune response, it will be elevated relative to normal, but it's never going to be like a huge percent of your blood because the job of white blood cells is to be out in the tissues fighting pathogens. So after they have fought whatever pathogen it is for their death and removal, most of them are gobbled up by macrophages so that they get removed from the system after they've done their job. In the case of neutrophils, they're the major component of pus, so one of the ways that you rid them is they excrete that onto the epithelial surface. So for their removal, we can say after fighting pathogen, they get eaten up or phagocytized. We'll say after fighting pathogen, they're removed by macrophages. And we'll go through all the really nitty-gritty details when we get to the immune system. Questions on leukopoiesis? All right, thrombopoiesis is the process by which we get platelets. And thrombopoiesis begins with the same hematopoietic stem cell, but it's going to follow differentiation pathways to become what's called a megakaryocyte. So our hematopoietic stem cell differentiates to become what's called a megakaryocyte. So this again is happening in red bone marrow. All of our blood production happens in red bone marrow. And red bone marrow is found within spongy bone. And if you remember the structure of spongy bone from 201, it's kind of organized in these trabeculae. And so if we were to look at a megakaryocyte, there are these really big cells that get these little things called pseudopods. And the pseudopods will stick out between sections of the bone, and the blood flow going past will shear off these pieces that become proplatelets. So our blood flow going past is going to shear off little proplatelets that become platelets, and then they go about and do their job of circulating around, looking for breaks in blood vessels and then forming that platelet plug. So we'll say they start as a megakaryocyte. The megakaryocyte sticks its little feet through the bone. So they're like pseudopods, fake feet. So we'll say sticks, pseudopods through bone. And blood flow shears off our proplatelets. Proplatelets become platelets. They are just cell fragments, so they only last about 10 days, or they last until they are used up in the clot. So they become platelets that circulate for about 10 days, or until we have a clot. After 10 days, they'll be filtered out in the liver and the spleen. So that's how we get all of our formed elements. Erythropoiesis gives us red blood cells, leukopoiesis gives us white blood cells, and thrombopoiesis gives us platelets. Any questions? Or until we have a clot. So they circulate for about 10 days or until we have a clot, After 10 days, they're not really good anymore. It's just cell fragments, so the liver or the spleen will take them out. All right. Hemostasis is the stoppage of bleeding. And it's very important to be able to do hemostasis quickly. If you've got a damaged blood vessel, we have to stop it from bleeding very quickly. So there are 3 steps in hemostasis. The first is called vascular spasm. And this will happen in the damaged blood vessel. So the damaged blood vessel will constrict to prevent blood loss from that blood vessel. So the first response to a break in a blood vessel is going to be vascular spasm, when this is a really prompt constriction of the damaged blood vessel. The next step is called platelet plug formation, and this occurs as platelets stick to the collagen fibers first. So platelets will first stick to the collagen fibers that are exposed in the damaged vessels. And then they're going to secrete these chemostatic factors and that are going to draw other platelets to the area, and they'll start sticking to each other. So then we'll have more platelets get pulled from the blood and stick to each other. So this is a little positive feedback loop that happens where more platelets present are going to attract more platelets and become sticky, make the other platelets sticky, so we get more platelets. It doesn't last for very long, but more platelets arrive and stick to each other. And we'll form a platelet plug. So this is going to be the second step, which will prevent some blood loss from that damaged blood vessel. But it's not very long-lasting, it's not very good. So the last phase is going to be our most potent mechanism of hemostasis, and that's coagulation. So the last phase is the coagulation phase. Coagulation is a pretty complex process. There's an intrinsic pathway and an extrinsic pathway. I'm not going to test you over the details of it because it's just too much. You guys have enough to worry about, but this is the most powerful mechanism of hemostasis. Oh, that says mechanism. And what happens in coagulation is we're going to pull that soluble fibrinogen out of the blood, and it's going to be converted to the insoluble fibrin. So remember we said one of the proteins floating around in blood plasma is fibrinogen. Ogen means to create, and it's going to create fibrin. So we'll say fibrinogen is pulled from the blood and converted to fibrin. Fibrin is an insoluble protein that's going to trap all the platelets there in the clot. If there are any blood cells right there, they'll get trapped in the clot. So fibrin traps platelets and blood cells. In the clot. And that will be what ultimately blocks up the damage in the vessel and stops the bleeding. So coagulation is very potent, and it's going to be the best way to stop bleeding. after we're done with the clot, so the platelets will be secreting those growth factors that will help repair the tissue. After we're done with the clot, we've got to eliminate it. So elimination of the clot happens as a two-part process. The first part is retraction. And in retraction, the fibrin is going to squeeze together and pull the clot together. So it will, as that happens, it's going to squeeze all those chemicals out of the platelets. We said one of the things that platelets have are enzymes that help to break down the clot. So this retraction happens as the clot contracts and squeezes on our platelets. So they will release enzymes that are going to help with the next part, which is fibrinolysis. So lysis is always breaking down, so we're breaking down fibrin, and these enzymes are going to help with that.